So maybe to close us out, I can show you guys a few links from some of the data resources that that I've been developing. So just a second or two, because I know we did pass the hour. But so at NeMO Analytics, which is a public website that's up and running, we have some really big collections of data. So for instance, I just typed in SYNGAP1 here, and this is a collection of human in vivo neocortical data. And so each one of these plots is an entire matrix of data from, in this case, portions of in vivo data. This is the Baduri 2021 aerialization study. This is their V1 PFC. And we can see here, at least in this data, SYNGAP1 is clearly in the neurons. If I zoom in a little bit, we can see there is sparse staining back here, not staining. This is RNA, single cell RNA. In other places, but I don't want to get into one data set too much here, just to show you that if you want to go to these websites, you can see basically any study ever done in the neocortex for gene expression. You can see where any gene of interest is expressed. This is the Poliudakis study that you used, Aditi, in the first talk today. This is Trevino. These are some older ones. We also have the laser capture from the Allen Brain Institute and Wieland Huttner's group some time ago. Bulk RNA-seq. So here you can see, and this is probably associated with its synaptic role here, where during fetal life, its levels are going up, and across life, it sort of takes this long trajectory that's falling here. This dotted line here is birth, and this is bulk PFC data. We even have a little bit of proteomics data. So this is SYNGAP protein across fetal and early postnatal life here from the Kriegstein lab. So this is and I'll send these links to the email list. So if you're not on the email list, let me know. So this is human in vivo. Here's some mouse in vivo data. This is the Di Bella data from the Arlotta lab. This is the atlas that came from La Mano from Sten Linnerson's lab. Here again, it's pretty sparse, but definitely both in progenitors and in neurons later, expression is visible. And finally, we also have a big collection of in vitro data. So this is from the Usquiano, sort of very detailed time courses of organoids. We also have the Baduri cell stress study that you mentioned today as well, Aditi. So, just a very general tool, but its power really is that all the data sets are here. So any data set that you've ever seen, both in vivo or in vitro, is probably here. If it's not, let us know. And this is where you can just type in a single gene. But if you had a list of genes, say your 10 favorite genes that go up or down in a radial glial cell that is harboring SYNGAP mutations, you can upload gene lists, and then instead of displaying a single gene on this map, you could be looking at your composite gene signature for whatever you're interested in. So I'll send these links around. I just kind of wanted to make you guys aware that this is here, because we've spent a lot of time putting the data together, and we really like to let people use it. Like I said, it's totally functional and public already. We're writing it up as a paper, but that'll be some time coming. So yeah, I'll send those around. And thanks so much for the talks today, both not only for neural differentiation, but for differences in humans and individual mutations. These talks are really at the center of so much thinking that we, I think, are all in the middle of. So thanks so much, and see you in a month.